Okay, welcome everybody. Sorry about the delay in the beginning of the session. I'd like to introduce Christoph Lotz, who is an epigeneticist and a biochemist, and he's going to talk to us all about the field of space biology. Thank you so much for coming.
So, hello.
Hello.
So, we are all pretty excited about the era in we live right now, right? And I think this exciting time where we are on the brink to become a multi-celestial species actually is similar like the time when our old ancestors went over the sea and discovered new fields and everything was new for them. But they were still on the same planet. And they brought their food with them and maybe even some pets and animals, what they needed on the new fields. And we need to do the same thing in order to become a multi-celestial species. So we know all how we get there, right? We know Elon Musk and the big rocket and Mars, and we know him. So we know how we get there. And I see that in the whole discussion here, the whole discussion is always, okay, we need a rocket, we need a bigger rocket, or the big rocket is a problem. And so we know these things, but how we live there, that is actually kind of a more submerged problem. So we know how we want to live there, like here on the moon with this kind of 3D printed house, and that is one idea. Or I saw this picture a couple of times in the convention here, like a Mars station. But what does actually the Mars condition and the moon condition environment does with our body? That will be decided not by engineers, or will not be solved by engineers and entrepreneurs like Elon Musk, but by space biologists, or by biologists, microbiologists, and botanists actually. So first I want to talk a little bit about us in space. So what does space do with us? And the general thing, what we all probably know already, is the following thing. So there's the cosmic radiation and ionizing radiation increases the risk of neurodegenerative diseases, and it compromises the immune system. So that is the common stuff what astronauts experience. And due to the microgravity on the ISS, we have this fluid distribution is shifted to the upper body, and that causes actually the nasal congestion, and we cannot smell anymore, and the taste is different. And also because of that, we have the pressure in the skull, the intracranial pressure increases, and that causes actually damage and abnormalities to the eyes and can damage the eye nerve. And what we just heard in the talk, we have a loss of muscle mass and bone density, and because of the bone density loss, we have more calcium in our blood, and that causes also kidney stones. So we have already, or astronauts have already, a lot of problems in space. But there was recently, or not recently, last year in February, there was this big study, the twin study, with Scott and Mark Kelly. And that was for geneticists, was it actually pretty exciting, because that was an identical twin study, right? So all changes we could see in his body, we could compare that directly to the twin study, to his twin, and could relate that to his habitation in space. So there is not much data out right now, but I want to tell you what NASA released already. They returned last year in February, and in January this year they put out some data on nature.com, and I read the article, and they released only a few data, but I want to discuss that with you a little bit, because I think that is not so well known right now. So the first thing what they discovered, it's not really surprising that they have a different gene expression level. So that is not surprising because it's a very different environment. Sadly, they didn't say what kind of genes increased or decreased in activity and how much. I attended a little conference in UCSD and they really didn't say that. They wait for the big release for the papers in 5 to 6 months or so. But the next two, they are a little bit more surprising results. So Scott Kelly got a change in the telomere length. Actually, the telomeres I explained very quick. Telomeres are the ends of the chromosomes on each chromosome in the cell, and they are kind of related to the aging process. So older people have shorter telomeres than younger people. And after each cell division, the telomeres get a little bit shorter until you get the DNA is damaged and then the cell cannot survive anymore. So it's an aging-related issue, and Scott Kelly's telomeres got actually longer in space. And yeah, that sounds great, but NASA was really surprised, and they said, okay, we have no idea why that is, and we need more experiments. So they set up an experiment. They didn't say much in the article about that, and they expect data in 2018. The real funny thing is that after Scott Kelly returned back to Earth, the telomeres got to the same length like before. So that's kind of like the cell knew, okay, I'm actually only that old and I get short again. So that is a very surprising result. So that was one.
Significant. I mean, was that a significant change?
Yeah, I mean they said that in the article. There was no data, and we need to wait for the paper. That's sad, but yeah. So, and the third really nice result was that the overall genome methylation level decreased. And what that means, I want to explain very quick. That is an methylated cytosine is an epigenetic marker. So we have here the normal cytosine, that is one of the 4 bases in the DNA code. And the cytosine can become methylated. So, and why is that? Why can that experience? So when you think about that, you have here your DNA, that's the DNA, and then you have here a gene represented by this block. And in front of each gene you have a kind of a guidance sequence called promoter. And this promoter decides basically what gene is how active at one point in which cell and what point of your development. And not all, but many genes have a so-called CPG island. A CPG island in this promoter region. And the CPG island consists of cytosine and guanine. This is a little D nucleotide, and that is very enriched into these regions. So that's basically represented by these little bit mushrooms here. So, and when this CPG island is not methylated, like the picture before, the cytosine has no methylation, then the gene is potentially active. So transcription factors can bind and can read the gene. So, and when it's methylated, it's inactive. So it cannot really, the suppression is suppressed. The expression is suppressed. So Scott Kelly now experienced an overall demethylation. So all genes which were potentially inactive could potentially become active, and that is a kind of the thing when you not maybe in his condition right now, but when you think about a longer exposure to microgravity, we have a lot of viruses we picked up in the evolution, and they are demethylated, they are methylated and inactivated, so they could become potentially active again. And also in the development of an embryo, we need a very, very distinctive program, an epigenetic program, that the embryo grows in the right way. So when you think about that this could maybe also have an influence of the offspring in what we would have in space, that would be a very, very big influence of that. So now I want to talk a little bit of our companions in space. So what we need to bring actually is in this picture here. So we need basically plants for our oxygen. So I have here kind of cyanobacteria and tomatoes. So we need an oxygen regeneration system. Then we need food. We need food plants. So we can also buy this. And then we need a system what get rid of the organic waste, what mostly will be a system made of fungi and bacteria to get the sources back, to the resources back to the both other systems. We also, I speak about that, we can also produce biofuel with these bacteria. So first I want to speak about this system, very, very brief. There's a group at the, no, that's the plants. Sorry, I start with the plants. So about the plants, that was a question never occurred actually right here. We have that plants really cannot really grow on the ISS. I mean, they grow, but not very well. And there's a publication what suggests that the threshold for the gravity sensing of plants is around 0.3 G. That is very good for us because we all want to go to Mars, and it suggests that we can actually grow plants on Mars without many problems because the gravity on Mars is 0.38. So that is an estimation that we can grow plants on Mars. But on the Moon, the gravity is much lower, so we will have problems to grow plants on Mars, on the Moon. So the next thing what we have to think about is our bio-waste management. And there's a group at the DLR in Germany, and what they did, they had a simulated urine, and they tried to clean that with a bacteria biofilter, basically like that. And you have here a filter material what is inhabited by certain bacteria strains, and you put here the urine on top, and then it goes slowly through, and then you pump it up again, and to clean basically the urine, and can use it for plants and for other things. And what the problem was, you needed to dilute the urine a lot, and the rate when it was cleared up was actually very slow. So this system, or these systems in general, have to be improved before we go to Mars. So then there is a suggestion that we can use cyanobacteria for a lot of things on Mars. So we can use it for oxygen recycling, also for food, and also for the biofuel production. And here's a kind of a picture of this kind of Mars colony. So we would have here a cyanobacteria pool energized by the solar energy. Here we have here another one. We can use regularly to have some nutrients, and then we can produce a biofuel. So why we need biofuel? One is for the rockets, but we need also biofuel for, at least at the beginning when we have no nuclear power plant over there, we need some engines to drive around, because nobody mentioned that, but Mars gets only 44% of the solar energy, so compared to Earth. So solar energy alone as an energy source on Mars is not really a good option, and will be not sufficient enough. So there I want to propose actually. So that these are the problems. So cyanobacteria need solar energy, and they will, when they are not heavily genetically engineered, they will have a problem to grow efficiently. So growing cyanobacteria for biofuel is actually even less efficient than growing cyanobacteria on Mars. So I want to propose a new organism for this purpose here, and this is actually a slime mold. So it's Physarum polycephalum, and that this slime mold is actually one single cell that has no cell walls and depends on.
Dictyostelium?
No, that is the other one. That is a true slime mold. Has no cell divisions, or has cell divisions, many nuclei, but no cell walls. So it's one single cell basically, and it lives from decaying organic material. So we can use that actually for decaying our plant material. So just a quick update. So this was the stage what we saw, and then it can goes into a sporulation phase. So we have here a sporulation phase, and then depending on the habitat, you have little ammoid cells or flagell gametes, and they can also interchangeable, and then they fertilize together and form again these things. So, and it's kind of intelligent, and people think about this organism to use it as a kind of biocomputer, because when you fill up a labyrinth with this slime mold and put here a food source and here a food source, it's able to find the shortest path through to the labyrinth. And also when you put food sources at the main train stations in Tokyo, it is kind of able to recreate the train system, because it's very efficient in finding the shortest path. So this is a very interesting organism anyways, but we can use it for this. So why we can use it? Because they figured out in 2012 already that this slime mold is actually kind of perfect to make biodiesel. So we can use that on Mars. I know we need also oxygen for these engines, but and it can grow on very easy carbon sources like rice bran. So you can use other sources we have on Mars then for our growth. So the thing is, the advantages are perfect lipid composition to produce that. It's very easy to harvest the lipids, because when you have other microalgae or so, you have a cell wall, and these are hard to crack and to crack open, and you need a lot of energy for that. So this organism doesn't have cell walls, just a cell membrane. So it doesn't need light. We can grow that on the waste material what we have already, and it's radiation resistant. I actually tested that myself back in my day. So against gamma rays, it doesn't care about at least that. I don't know about cosmic radiation, but ionizing radiation doesn't care. And it can be cultured in a bioreactor setting. So my conclusion is, when we go to Mars and so on, we need to figure out first. We need to deepen more the microgravity research. So what will happen to us, to the plants, what we bring into the bacteria. And we need to find biological solutions for that, what we can actually bring, because technological, yeah, we can do that, but biology works better. We need to adapt that for these findings, maybe genetically and also other ways. So, yeah, and we need to adapt us and our companions for this kind of matter. So, thank you very much. Yeah, questions?
How quickly can that mold consume what quantities of material? I mean, I'm thinking you process a fair amount of waste.
Yeah. I think that is a good question. I'm not sure about that, but in the biofermenter you actually don't use the whole flat fungus. You use micro. You basically. You can cut it in very tiny bubbles, so you have a much bigger surface. So you can have probably a bigger amount. But I don't know a rate actually. But that's a good question. I looked that up.
And is there no cellulose? Can it eat mostly cellulose?
Most what?
Cellulose.
Cellulose. So you mean materials or kind of? All decaying organic material. It lives basically in the woods and eats mostly everything. Yeah, you can grow that on oats and yeah.
What's the best estimate as to why that organism is so resistant to radiation?
I think that is kind of, that also a good question. I don't know for sure, but there is this other organism that is a bacteria, what is Deinococcus radiodurans, and this has a few copies of its genome in the cell. So, and it can kind of intersect, okay, this is damaged, so, and I can repair that much easier. So, and because this has thousands of nuclei in it, so maybe some get damaged, and then you can, slowly, but that's a good question. I don't know. Maybe there's no reason why it's resistant against radiation. But
So does it mostly have the ability to do a lot more DNA repair than most other animals?
That's definitely a good research project for that organism. Sadly, I need to drop that project before. There are maybe different repair enzymes. Yeah. Oh.
Last question?
Did you mention the organism? Quick question.
Did I mention the
What the organism is?
that? What?
What's the name of the organism?
Oh, Physarum polycephalum. No, what do you mean? It's this.
It's in the abstract. It's in the abstract. Okay, I'm sorry.
Abstract. Oh, did I? It's in the abstract. Yes. Oh, I'm sorry. Yeah. Okay. What he was?
Yeah, just a question. Why would we need to study twins to figure out the differences in their epigenetic profile? How do we just study one individual before going after?
Because each human being is actually, when it comes to these things, very, very different. I mean, look at that. They are very good athletes. They are not good athletes. So that is already a different genetic set. And when you put one when you compare these people in different environments, it's not really comparable. You cannot really compare that. I mean, you can compare that, but you need then a large number of. Okay. Yeah. Thank you.
Okay, well, thank you. Thank you for